Hi everyone, here we are with another lecture video. Today we're going to be talking about membrane transport, diffusion and osmosis. This topic comes up in Bio 101 when we talk about the membrane, the cell membrane, and its function, its structure and function. It also comes up in microbiology when we talk about cell structures and their function. And again, when we talk about cell growth and control of growth, so chapters 6 and 9. This also comes up in chemistry and physics, in most biology classes that have to do with cells. It's because cellular or membrane transport is how things get in and out of the cell, so it's pretty important. And also because students struggle with it. And so it's important to keep going back to it and double checking the concepts and the vocabulary. Some of the vocabulary, conceptually, it's pretty easy, but the vocabulary is where students tend to trip up. So we're going to walk through that. What we'll talk about first is just very quickly membrane structure and function, what it's composed of, just a quick review of what it's made of and how those components work. And then we're going to spend most of the time talking about passive transport across the membrane, and specifically diffusion and osmosis, and the terms that we use to determine which way water is going to move into or out of a cell. This is important for cells maintaining their functionality, maintaining homeostasis with water balance. You will see what happens to cells if they can't do that. Before we start, we're going to do a little experiment while I talk. So here's a glass of water, and I have some food coloring. And I'm going to put a few drops of food coloring into the water. And you see that the food coloring is kind of all clumped. It's highly concentrated right along the bottom. All that food coloring's sunk down here in the glass. So I have a high concentration of food coloring here, less concentrated here, sort of mixing in, but more toward the bottom of the glass. So higher concentration. So this is an experiment. So let's come up with a hypothesis. My observation is that the blue food coloring molecules are concentrated toward the bottom. What's your hypothesis? What's going to happen over time in this glass if I just leave it to sit here? So from experience, you might know that eventually the blue will be distributed evenly throughout the water in the glass. This is an example of diffusion. Molecules move from an area of high concentration to lower concentration. So over time, I expect the color to become uniform throughout as the molecules move through the glass. So we'll come back to that and see if that's what happens in a little bit. So onto our plasma membrane. So my plasma membrane, all cells have a plasma membrane or a cell membrane. Don't confuse that with the cell wall, two different things. Bacteria have a peptidoglycan cell wall. Plants have a cellulose cell wall. Fungus have a chitin cell wall. And so different molecules, but the cell wall is just a rigid structure that gives the cells shape. It doesn't have any metabolic function. The plasma membrane is a metabolically active part of the cell that controls what comes into or leaves the cell. So it's what separates the inside of my cell, my cytoplasm, or cytosol. So that prefix cyto means cell. And sol, if we call it cytosol, it's because it's a solution in there. It's stuff dissolved in water inside the cell. From what's outside the cell. The plasma membrane can be the site of chemical reactions in cells. And its fluid mosaic model, this is what we call the model of the cell, is phospholipid bilayer with embedded proteins. So we'll look at that structure. The three components of the plasma membrane are that phospholipid bilayer with proteins that are floating around in the phospholipids. So the phospholipid bilayer has the consistency of olive oil, and those proteins are kind of floating around through there like icebergs. Some of those proteins run all the way through, extend from one side to the other of the membrane, and some are just sitting on one side or the other. And then membranes have some sterol in them that's function is to maintain the consistency or the fluidity of the membrane. In animal cells, that's cholesterol. It's different sterols in different types of cells. And that's just to make sure that as temperature changes, as conditions change, that membrane has the same consistency all the time, that olive oil consistency with those proteins able to float through. So here is my fluid mosaic model. So a mosaic is a picture made of tiles. So the fluid would be the phospholipids that have the consistency of olive oil. And the mosaic, the tiles, would be the proteins that are scattered throughout and floating throughout. Here's a close-up. I have my phospholipid bilayer. It's a bilayer because this glycerol head on my phospholipids is hydrophilic. It loves water, so it wants to be touching the water. And I have an aqueous solution inside and outside the cell, so those heads orient to in and outside, while the hydrophobic fatty acid tails want to get away from the water, and so they turn in, giving me my bilayer structure with embedded cholesterol. So this is an animal cell, and proteins that can be embedded or on the surface. Those proteins, there are 6 primary types of proteins that we find in our plasma membrane that have different functions. The first are channel proteins. These basically act like a doorway through the membrane to allow molecules to pass in and out. The molecules that are going to use these channels are things that are too big to squeeze in between the phospholipids, or they could be molecules that have polarity. So if they are charged, an ion, for instance, or something with polarity, they would get sort of stuck up or caught up or repelled by those hydrophilic heads, or get caught stuck in the middle of those hydrophobic tails. And so they don't pass readily through the phospholipids, even if they're small enough. Then I have carrier proteins, which are similar in that they provide a doorway, but they are the specific shape to a specific molecule. They can also work in combination with ATP to actively pump or push those specific molecules in or out of the cell. I have cell recognition proteins. This is how our immune system recognizes cells that belong versus cells that don't belong. Receptor proteins that have a specific shape that allows certain molecules to attach or bind. Some examples are receptor proteins for hormones. So hormones only act on specific cells. They're the exact right shape to fit into these receptor proteins to trigger some sort of reaction in the cell. We have enzymatic proteins. These are proteins that carry out metabolic reactions. In bacteria, where I don't have membrane-bound organelles, most chemical reactions are going to take place on the plasma membrane, on the cell membrane, at these enzymatic proteins. And then the last are for multicellular organisms that have the tissue level of organization, how cells attach to become tissues and how they can communicate with each other is through these junction proteins. Just a closer look at some that we will encounter a little bit later in microbiology when we talk about the immune system. We'll look at cell recognition proteins, specifically the major histocompatibility complex. These are a series of glycoproteins, so protein with a sugar attached, that are specific to each person. They're different for everyone. That's why we reject organ transplants. Our immune system runs around, finds this, says, oh, you're me. Oh, you're me. When it sees one that's different, it says, oh, you are not me. You don't belong here, and attacks. So recognition proteins let our immune system see what belongs and what does not. Talk about receptor proteins that can receive molecules, chemical signals within our body. For viruses to gain entry into a cell, many of them use receptor proteins. Their external spikes have to encounter cells that have a receptor protein that they fit into. That's why viruses target specific cells. So respiratory viruses have protein spikes that fit exactly into the cell surface protein on their target respiratory tract cells. And then enzymatic proteins, these are going to catalyze specific chemical reactions. So here's a reaction where here's my reactant. It lands on the active site to form the enzyme substrate complex, and the chemical reaction is turning it into this molecule. This is just a summary of the 6 different types of proteins. These are the 3 that we talked about just then in more detail. These are my 2 doorways, channel proteins. If you can fit through, you can go through. Carrier proteins are a specific shape for a specific molecule. And then my junctions to give me a way for cells to attach or communicate with each other. So the function of the plasma membrane is to control what enters or leaves a cell. It allows some to move freely and others it prevents from moving freely, either in or out. Small molecules, as I said, that are nonpolar, oxygen and carbon dioxide, there we go, can pass through the phospholipid bilayer freely. They will move from an area of high concentration to low concentration, so down the concentration gradient. Think about the word gradient means a slope. So moving from up here on the slope, high concentration, to down here, low concentration. Molecules do that passively, takes no energy. I always think of this about as riding a bike. To get from high to low, I don't need to add energy. This is going to happen spontaneously. The energy contained within the atoms of those molecules gives them some vibratory energy, and that's how they move, and they spread from high concentration to low. Like we see happening here, we still have a higher concentration toward the bottom, but we are getting toward a more uniform color. I'll give that some more time. Water also moves across the plasma membrane. Water has some polarity, if you remember. Our water has slight polarity, slightly negative around the oxygen. That's a bit of an electron hog. And so it can pass in between those, it's small enough to pass in between the phospholipids, but because of that slight charge, it moves really slowly. And when the cells are trying to balance water, it has to happen at a pace that can allow them to maintain homeostasis. And so we have a special set of proteins that are called aquaporins, and those specifically let water move in and out. So those are all passive mechanisms. Is going to have water moving across from high concentration to low, either right across the phospholipids, through carrier proteins. Either way, it's passive. If I'm talking about any molecule at all, it's diffusion. If I'm talking about water, we call that osmosis. We'll get to those terms in a minute. And I also have active transport. Active transport means energy is required. That can be through a carrier protein with the addition of ATP, moving against a concentration gradient. So here's my slope, which means my slope that way. And I'm down here, low concentration. To get to high, I have to go uphill. That means I'm going to have to pedal if I use that bike analogy. I'm going to have to put in energy, ATP. It can also involve bulk transport, where I use transport vesicles formed of plasma membrane. So I can take my membrane and I can fold it around something to bring it into the cell, endocytosis, or I can use one of those transport vesicles, fuse it with the membrane to let something out of the cell, exocytosis. So we talk about the membrane being selectively permeable. It can determine what gets in and out. Here we are. That can happen passively for molecules going from high concentration to low concentration through the diffusion of molecules across their concentration gradient. Osmosis, specifically the diffusion of water across a membrane. So this is a special case of diffusion. Facilitated transport, I'm going to use one of those proteins to let molecules move down their concentration gradient. And then active transport, where I need to add energy or ATP to move against a concentration gradient, or because I'm transporting in bulk and I have to move transport vesicles, so that requires energy. And I use those usually with bigger molecules. So if we look closely at how different molecules can cross the membrane, here's my phospholipid bilayer with some proteins embedded. I've got aqueous solution, cytosol or cytoplasm on the inside. I've got an aqueous solution on the outside. Small charged molecules or ions that have a charge are going to be repelled by the glycerol heads, so they can't cross the membrane. They're going to have to go through a special protein, through one of those channel proteins. Water has its special protein, its aquaporin, to go through. Non-charged molecules that are small enough to fit through here can pass right through the phospholipid bilayer. If they're very large, they won't be able to get through, so they're also going to need a specialized protein doorway. So my ions and charged molecules and things that are too big will need either a specialized doorway, or if it's a very big macromolecule, we might use endocytosis to get it in, or exocytosis to get it out. Wrap it in a vesicle. So passive transport, when we're talking about passive transport, we have to look at what's inside and what's outside to figure out what that concentration gradient, where is the concentration high, where is it low. So we're going to compare two solutions. And a solution consists of a solvent, some liquid, and a solute, a solid that's dissolved in the liquid. In biology, water is our universal solvent, so we're talking about water when we talk about a solvent. Diffusion is the net movement of molecules down a concentration gradient. So net movement is from high concentration to low. So molecules can move both ways. So even though this is where the concentration is higher, molecules as a whole of food coloring are moving to the lower concentration up here. But it's not a one way. Every now and then a food coloring molecule might move down into here. So we're not only moving one way, but the net movement, the overall total movement, is from high concentration to low concentration until equilibrium is reached. And equilibrium is all of the molecules are evenly spread. The solute concentration is uniform. There is no gradient. There's nowhere with a higher concentration. We think that's what we're going to see here. Right now, my food coloring had been in the refrigerator, so it's colder, and temperature, pressure, electrical currents, molecular size, those can affect the rate of diffusion. So diffusion is probably happening pretty slowly now while I have this temperature difference. But as the temperature becomes uniform throughout, those molecules will start to move a little bit faster. And we're going to move until we reach equilibrium, until all the molecules are evenly distributed. There's no gradient. So let's look closer at what we're talking about. So when we talk about a solution is composed of a solute plus a solvent. An example everybody's familiar with is Kool-Aid, which a pitcher of Kool-Aid is a solution made out of colored sugar, which is a solid that's dissolved in water. The solute determines the concentration. The concentration is the portion or the percentage of the total solution that is solute dissolved in water. So when I give a concentration, that's what I'm talking about. When we're looking at diffusion, what we should see here is I have this is crystal dye, so it's a solid, it's going to diffuse. Starts out highly concentrated here. All the molecules are here. All the water molecules are here. Over time, diffusion will cause those molecules to start to move to where they are in lower concentration. When I'm talking about diffusion, I can be talking about any molecule. In this case, I have dye diffusing to where there's a lower dye concentration. Here I've got a high water concentration. Here, hey, no water, low water concentration. Water is also diffusing to where the water concentration is lower. So I have the water and the dye both moving. They are both diffusing, and that's going to go on until the dye, the solute, is distributed evenly through the solvent, the water. That's equilibrium. All the molecules are evenly distributed. In that case, I didn't have a membrane, and we're talking about membrane transport. An example of diffusion for cells in our body is gas exchange, oxygen and carbon dioxide in the lungs. I inhale, I get lots of oxygen, high concentration in the lungs, and it diffuses into the blood vessel where there's a lower concentration. Carbon dioxide being carried back to the cells would diffuse out and be exhaled. So from high concentration to low. Back to my solution of Kool-Aid. So as I said, the concentration is determined by how much of this is dissolved in the water. So here I have two concentrations. I have 5% sugar. I have 95% water. I don't call this a 95% solution. The solution concentration is determined by this, my solute. So this would be a 5% solution. So solution, some portion is the solute and the remainder is the solvent or water. Here in my sugar packet, this is 100% solute. There is no solvent in the sugar. Here's my water, my solvent has 0 solute. So I would call that a 0% solution. There is no solute. There is nothing dissolved in this water. This is 100% water. Then if I pour the sugar into here, I get a solution. So in this case, I have water, which I know is a 0% concentration because it's water, and the concentration refers to how much stuff is dissolved in the water, whether that's sugar, salt, amino acids, anything, how much is dissolved. These brackets here, that's a chemistry way to indicate concentration so that I don't have to write that word out all the time. So pure water has nothing dissolved in it. It's 0%. So any other solution has a higher solute concentration. Even if it has a 0.0001 sugar concentration, it still has a higher solute concentration. This is the ultimate hypotonic solution. So we'll look at our tonicity terms. The tonicity refers to the relative concentration. Relative means compared to. This compared to that. So these terms for tonicity don't talk about what's the tonicity of this. I can't know the tonicity of one solution until I know what I'm comparing it to. I'm not comparing solute, my 5% sugar, to solvent, 95% water. That's just my concentration. Tonicity is relative concentration. This compared to that. I have to have two solutions that I'm comparing. So here I have 5% solution. Here I have a more dilute solution, 2%. So the picture on the left has a higher solute concentration. So we can use the tonicity term hypertonic to describe it. You think hyper, if you're hyper, you have high energy. Hypertonic solutions have more solute compared to another solution. 5% solute compared to 2% solute. This is hypertonic compared to that. If I'm comparing two things, then I can describe this one relative to this, or I can describe this relative to this. So here, I can also describe this picture with 2% concentration. This is lower than 5%. So I can describe this picture as being hypotonic compared to the one on the left, to 5%. So those tonicity terms, hypertonic and hypotonic, if I have two solutions, if one is higher, the other is lower. So relative terms mean I'm comparing. And if one is high, one is lower compared to the other. And so I can't just look at one solution because I don't know what I'm comparing to. Now, which solution here has the higher concentration? I'm comparing two solutions. Here, a 5% Kool-Aid solution to a 10% Kool-Aid solution. Now this picture has a lower solute concentration, so I can say it is hypotonic compared to this solution. This has more solutes dissolved in it, so this is hypertonic compared to this solution, which is hypotonic. So I can compare them one to the other. How about if I have the same solute concentration in both? Then we use the term isotonic. That prefix iso means the same. So iso bars on a weather map show areas with even pressure. So isotonic means I have the same concentration in two different solutions. The picture on the right is isotonic to the picture on the left. The picture on the left is isotonic to the picture on the right. So my tonicity terms, if I have isotonic solutions, the solute concentration in a solution over on this side is the same as the solute concentration on this side. They're both equal. There's no net movement. So here I have same concentration. There's no concentration gradient. My gradient has gone from here, high concentration to low, to an even slope. There's no gradient. That bike's going to stay still. It's equilibrium. There's no net gain or loss of water. Hypotonic, low. Think hypo is low. Hypotonic solutions have a lower solute concentration compared to what's on the other side of the membrane. If I put a cell in a hypotonic solution, then what that term is referring to, the solution out here is hypotonic, has a lower solute concentration than what else? Than the concentration inside the cell. So I'm going to compare either side of the membrane. So if this is hypotonic, has a lower solute concentration. If I have a lower solute concentration, the water concentration must be higher than in here. Water will flood into the hypertonic cell and the cell will burst. An animal cell would burst because typically, like one of our cells, if we put it in fresh water, which is the ultimate hypotonic solution, water would rush in trying to get to equilibrium, and eventually our plasma membrane would burst when the cell fills up. Some organisms, like protozoans that live in fresh water, would blow up all the time if they didn't have some way to balance that. Protozoans have contractile vacuoles to get rid of excess water so that they don't blow up. When I use these terms and I'm comparing the solution inside a cell to outside a cell, and I put my cell in a hypertonic solution where there's more dissolved substance than here. If there's more dissolved substance here than there is here, then that means there's less water here compared to here. So the concentration of solute is higher, hypertonic, in the solution than in the cell. If I put a cell into hypertonic solution, the water is going to leave the cell trying to reach equilibrium. And so the cell will shrink. That's called crenation in animals, or plasmolysis in plants. Because in animals, where we just have a plasma membrane, as we lose water, the whole cell shrinks. But in plants or in bacteria, the cell wall is going to remain intact because the membrane's inside, but the membrane's going to shrink.